everyone. Thank you very much, Eric and Laura. So I will talk to you about my project on insect visitors to Tarflower. So Tarflower, or Baharia racemosa, is a 4 to 10 foot tall shrub with a peak bloom in May to June. It's nearly Florida endemic, just a little bit of its range goes into Georgia, and it's pretty common in the Florida prairie scrub and pine flatwoods. Its seeds are in capsules, and it's the only shrub in the scrub with hairy stems, and it resprouts very quickly after fire. It gets its name from its sticky buds, petals, and fruit, and it's the only plant with sticky flowers in the Florida scrub. The stickiness comes from the glue that is exuded from between the sepals, and this coats the buds, outer petals, flower bases, and developing fruit. This glue traps many insects, and Eisner and Anna Shansley found that it is as strong as commercial insect trapping glue and can even trap large insects like June beetles and two-striped walking sticks, but it traps many other insects such as ants and flies. So the purpose of my study is to investigate these questions about insect visitors to tarflower. First one is, do sticky flowers inhibit crawling or flying insects? And then what insects visit tarflower and which ones are more likely to be stuck versus visit successfully and be potential pollinators? And then the third question was, what factors affect insect visitation? And I'll be talking about date, tarflower bloom density, and cloud cover. So for my methods, I looked at 322 flowers total that I observed, and in 42 patches. And I did 64 observations of those patches, so some patches were observed several times. And then I observed 2 to 10 flowers at a time in 19 days over 2 weeks. The day before each observation, I marked buds with string and put either sand on the base of the bud or no sand on the base of the bud. And the reason for that was the next day when the flowers opened, the sand on the base of the flower could let insects crawl over that usually sticky base of the flower and get to the nectar and pollen. And then the control with no sand on the base, insects would get stuck as usual. So for my flower visitor observations, they were each 15 minutes long, and before each one I took weather measurements. So I estimated cloud cover and then measured wind speed, temperature, and % relative humidity. And then during my observations, for each flower, I recorded the number of visitors that I observed and classified each of those visitors at least to family and many to genus. And then after my observations, I counted the number of other tarflower blooms within 2 meters of each bloom that I observed. So on to my results. I found no exclusively crawling insects on open flowers during my formal observations. And this picture shows the one ant that I found on the face of open tarflowers the whole summer. And in fact, most insects I found that were stuck on tarflowers were stuck on either the buds or some on the developing fruit, and not as many on the base of the flower like I expected. And the only crawling arthropods that I found on the open flowers commonly were spiders, and they were present with or without the sand treatment, so they could walk over those sticky petals without a problem. Crab spiders were very common, and I also found some green lynx spiders. So overall, here's the effect of covering up the sticky flower base with sand on various flower visitors. I found no evidence of effect on crawling insects, since there were none on the face of open, sandy, or sticky flowers at all. So the sticky flowers may be very good at inhibiting them. For flying insects, This graph shows that there is just a slightly higher mean number of flying visitors per flower per observation period on flowers with sand than those with no sand, but no statistically significant difference between the two groups demonstrated by the standard error bars on the graph. And as you can see from my y-axis, I have a pretty small visitation rate. I'm only seeing about 1 to 2 flying insect visitors per 15 minutes. And I didn't collect data on spiders, but they were on the flower with or without sand. So to sum it up, the effects of tarflower stickiness on visitors, I found many insects stuck to buds and fruit, and some on flowers in my casual observations. Lots of love bugs and other insects like flies, ants, and some other larger ones too. Many insects were less affected, including some moths and wasps that visited flowers right after the petals fell, but still could get some nectar without risking getting stuck to those sticky petals. Crab and lynx spiders walked over the sticky petals without problems. And amazingly, bees, except for honeybees, which are non-native, were almost never stuck. So this gets me to my second question, which is what insects successfully visit tarflowers? So overall, out of the 361 insects observed in those 19 days, bees were by far the most common at 93.8% of all visitors that I saw. There were 3 bee groups that were very common, the Megachilae, the leafcutter and resin bees, the Halictidae or sweat bees, and the Bombus or bumblebees. There are also some other insects that may use the tarflower as a resource, like some wasps, some other bees, and some butterflies, and a few flies. So now to my last question, what affects insect visitation? So I will be talking about date, tarflower bloom density, and percent cloud cover. So to investigate which of these factors are most important for predicting insect visitation, I built statistical models in R, which is an open source statistical modeling language. And I used negative binomial regression models, which are made for count data, which I had, and with XX zeros, and in my case, that means observations where I didn't see any insects, and more variation than expected. And I made models with combinations of these 3 variables, date, tarflower, bloom density, and cloud cover. And I did this to find variables that best predict the, or most affect, total insect visitation and visitation of the 3 most common bee groups, the Megachilae, Halictidae, and Bombus. And I used AIC values to choose the best models. So here's how I built my models and chose the best one using AIC values, using the example of total insect visitors. So with AIC, the model with the lowest AIC value is the best model. So this table shows the models I made and their AIC values. And for reference, the null model has no variables and has the highest AIC value. The models with only 1 variable, in this case, have slightly lower AIC values, so they do improve the model somewhat. The next 3 models, next models have combinations of 2 variables, and many of these AIC values are lower still. And in this case, the model with the lowest AIC and the best model is the one with all 3 variables. However, the AIC value isn't quite low enough to make it significantly different from the model with just cloud cover and bloom density. So then I looked at a summary table of this best model and found that the variable that most strongly affected total insect visitation was cloud cover. So next, I will take a closer look at the effect of cloud cover on total insect visitation. So this graph shows percent cloud cover on the x-axis and average number of total insect visitors per flower per 15 minutes on the y-axis. And as you can see, I observed more visitors when it is sunny and fewer when it is overcast. So here is what I found are the best models for predicting insect visitation based on their AIC values. The variables listed first have the largest and the strongest effects. So as I just discussed, total insect visitation is most clearly predicted by cloud cover, but bloom density and date are important as well. And interestingly, visitation by all of the three most common bee groups, the Bombus, Megachilidae, and Halictidae, were best predicted by models with date and tarflower bloom density. So it's very interesting that these three different groups are affected by the same two variables. However, each of these three bee groups are affected differently by each of the variables. So next, I will explain how each of the bee groups are affected by date and bloom density more closely. So first, let's look at bombus or bumblebees. I was curious about why all bumblebee observations occurred in the first week of my study. So I compared the graph at the bottom, which shows bumblebee observations over my study period, to the graph at the top, which shows the number of tarflower blooms within 2 meters of each flower that I observed. So essentially flower density over my sampling period. Flower density decreased with time, which makes sense since I did my study towards the end of the peak blooming season. So it is possible that bumblebees prefer higher flower density since they are large, efficient pollinators and need lots of nectar and pollen. Also, towards the end of my sampling period, I started seeing bumblebees visiting other flowers that were more abundant than tarflower at the time, such as Lyonia fruticosa. So this graph shows Megachilidae or leafcutter and resin bee visits and tarflower bloom density over time. There were more Megachilidae visits with lower bloom density, but the Megachilidae were present throughout the study. So one explanation for this counterintuitive finding is that Megachilidae may be more concentrated on the few plants still blooming at the end of the blooming peak, instead of spread out on the many blooming tarflowers at peak bloom. So this graph shows Halictidae, or sweat bee visits, and bloom density over time. There's a large increase in sweat bee visits in the last week of my sampling period, and one possible reason for this increase is I started seeing brown-winged striped sweat bees in the last week. These bees are much larger than the smaller sweat bees that I saw in the rest of my study. So they would likely be able to visit more flowers, and I might be more likely to observe them. So on to conclusions. So overall, shifts in potential pollinator abundance were primarily affected by date and tarflower bloom density. So as you can see from this diagram, bloom density decreased at the end of the blooming peak. So this may mean that plants that bloom where there's a high flower density are dominantly visited by larger, more efficient pollinators such as bumblebees. But plants that bloom towards the end of the blooming peak will likely be visited primarily by smaller bees, like the Megachilidae and the Halictidae. So while the density of tarflower blooms is certainly affecting bee families, very likely these pollinator abundances are also affecting tarflower reproductive success. I will investigate this question with the seed set data that I have collected and am still analyzing. So thank you to so many people for help with this project. Thank you to the Archbold Plant Ecology Lab and especially Eric Menges, the Vaughn-Jordan Foundation for providing funding for my internship, and all the other Archbold employees who helped out, and all of you for listening. And I can answer some questions if there's time.
Thank you, Lily. We have several questions. Carolyn Lanker asked, how does the stickiness benefit the plant?
So that is the big question, and it's still not completely answered, but one of the most likely hypothesis scenarios is that it could stop insects or other animals from eating the buds or the developing fruit or the flowers. Or it could also stop insects from stealing nectar or pollen, possibly crawling insects that would be less effective at pollinating than flying insects. It still hasn't been answered though. That was one of the things that got me interested in it.
Okay, and our former intern, John Benning, has kind of a two-part question. He wonders if you observed any spiders preying on the stuck insects, and whether you wanted to talk about whether the stuck insects would attract spiders, which would then maybe protect the plant from insect herbivores.
I did not observe any spiders preying on the stuck insects. The spiders that I saw mostly were very small, those crab spiders. They were even scared off by the small sweat bees. So it would have to be pretty small insects that were stuck that they would prey on. I could definitely see the link spiders eating stuck insects though. That's a possibility, I don't know. Yeah.
Okay, and then Scott asked whether you ever observed aggression between different families of bees.
So yeah, that is definitely, so one thing that I was interested in is could there be competition between these very large bumblebees and the smaller bees? I didn't ever observe aggression between them, but the bumblebees were much more abundant in the beginning, and the smaller bees were less abundant on the flowers that I observed. So it's possible that the bumblebees, even if they weren't like specifically attacking the smaller bees, the smaller bees might have just stayed out of their way because they're large. Yeah, I tried seeing if there was a basically doing the same analyses that I did with the entire data set on looking at the smaller bees and seeing if they were still affected by date and nearby blooms the same way after the last bumblebee observation basically. And I didn't find any change, so I didn't find any evidence of competition between those, but it could still be happening.
Okay, this is a related question from Hillary about Bombus. Are they so efficient that in that way they're outcompeting the megachilas and the Halictidae?
Yeah, so they are very efficient, and they if there is enough blooms there that the bumblebees are visiting the flowers, then they will be really good and visit like every single flower in a patch and then spread that pollen all over the landscape. But from what I found, it seems that they could be outcompeting the smaller bees in some circumstances. But from what I found, I think the smaller bees are still important in when there's possibly a lower flower density. They would still be visiting the tarflowers even when there's not as many flowers, whereas the bumblebees might stop visiting if there's not enough of that resource there to keep them keep this very efficient pollinator visiting the plant. So I think both the larger pollinators and the smaller pollinators are important for the plant. But they could.
That's a really interesting answer. I like that interpretation. We also asked if you looked at Mark Darab's flower visitor data to see if he observed the same trends.
Yeah. Oh, so I did look at Mark Darab's visitor data for multiple things. So he observed a majority of bees for sure, and also a large number of those same most common bee groups that I observed. He also observed some more flies, or he was doing collections. So there's a slightly different sampling method, but he collected more smaller bee flies, a few of them, and I didn't really find those. Otherwise, we were finding very similar visitors. And then I looked to see if there was any, if the bumblebees were visiting like flowers in like clumps, kind of like, let's see, if they tended to visit one type of flower and then if that got too few, then they would switch to another type of flower. And then in some cases they did, but it was unclear. Was that what you're asking?
Okay, there's a few more
Mostly
questions
similar.
that we'll have to save for later, Lily. But thank you very much, and I'll turn it back to Laura.
Thank you.